Hi everybody, today we're going to talk about meiosis, and meiosis is a special type of cell division that reduces the chromosome number from diploid, like us, where we have 2 copies of each chromosome, to haploid, which is what our egg and sperm cells are, or our gametes that we need to make the next generation. All right, so quick review of vocabulary. Diploid is when you've got 2 copies of each chromosome. These are your homologous chromosomes, so they have the same genes, just different versions of them. You've got 1 copy from your mom and 1 copy from your dad. Haploid is when you have only 1 copy of each chromosome in a particular cell. So all of your gametes, all of your sex cells are haploid. Right, so the function of meiosis is to produce these haploid gametes. And in order to do it, the cells have to do 1 round of DNA replication and followed by 2 rounds of cell division. So that's what we're going to focus on and talk about for the rest of the day, is how we do that replication and those 2 rounds of division. All right, so here is our starting cell. So this is a diploid cell. So we know that this cell is diploid because it has 2 copies of each chromosome. All right, so it has 2 copies of each chromosome. Here is chromosome number 1. And we have 2 copies, the red one from mom and the blue one from dad. And then the second smaller chromosome is chromosome 2. And again, we have 2 copies, one from mom and one from dad. Inside of your cells, your chromosomes are numbered. You have 23 pairs of chromosomes, and they're numbered in the order of size. So the biggest one is chromosome number 1. Your smallest one is chromosome number 22. All right, your 23rd pair of chromosomes are going to be your X and Y chromosomes, depending on whether you're male or female. Okay, so a couple other things I want to point out on this picture. So here, chromosome 1, we have 2 copies of it. These 2 copies have the same genes on them. So let's imagine you've got mom's eye color gene right here and dad's eye color gene right there. Mom and dad may have different colored eyes, so they may have different versions of the gene, just like the chromosomes are different colors, but the gene for eye color is in the same place on both chromosomes. And because of that, this pair are called homologous chromosomes. Okay, so on this diagram, we've got a total of 4 chromosomes, and these are coming in pairs, so they're 2 pairs of homologous chromosomes. All right, and now what I already told you was that in order to do the process of meiosis, we're first going to have to do 1 round of DNA replication, followed by 2 rounds of cell division. So let's talk about that DNA replication first. So during DNA replication, we're going to duplicate each of these chromosomes. So chromosome number 1 is going to get duplicated, both mom's version and dad's version. And chromosome number 2 is going to get duplicated as well, both dad's version and mom's version. All right, now these duplicated chromosomes are held together at the middle by the centromeres, just like we saw in mitosis. And so these 2 pairs of chromosomes, so the red and the blue are a pair of homologous chromosomes. The 2 copies of the red are called a pair of sister chromatids. Right? Sister chromatids are identical, so they have exactly the same genetic information on both copies because the second copy was made from the first copy during the process of DNA replication. So in S phase of the cell cycle, the DNA was then duplicated, making these identical sister chromatids. Okay, so this cell here is also diploid. So this cell is also diploid. It also has 2 pairs of homologous chromosomes. Let me phrase that again. So this cell is diploid. It has 4 chromosomes, just like we saw in the other cell. So it has the same amount of genetic information as we saw in the other cell. It just happens to have 2 copies of it. So now we're going to call these 2 copies chromatids. So it has 4 chromosomes and 8 chromatid. Right, so this cell here still has 4 chromosomes. They've now been duplicated, so now we have 8 chromatids, whereas before we just had 4. And now we are able to go through the process of meiosis. So we're going to go through 2 rounds of cell division. So the first round of cell division is going to be called meiosis 1. And in meiosis 1, what is going to happen is the homologous chromosomes are going to separate. So what we're going to wind up with is we're going to wind up with one daughter cell that has 1 copy of chromosome 1 and 1 copy of chromosome 2. And the other daughter cell is going to have a different copy of chromosome 1 and a different copy of chromosome 2. All right, so each of these 2 cells that we have here are going to have 2 chromosomes because they have 1 copy of chromosome 1 and 1 copy of chromosome 2. And in this case, those 2 chromosomes is going to equal a total of 4 chromatids. Right? So technically, these cells pictured here at the end of meiosis 1 are haploid because they have only one version of each gene. Right? Don't worry too much about that. After we do meiosis 1, we're going to have to do another round of cell division as well. This round of cell division is called meiosis 2 because it happened second. And in meiosis 2, the sister chromatids separate. And so what we're going to get at the end of meiosis 2 is we're going to get one cell has a copy of mom's chromosome 1 and dad's chromosome 2. The other cell has a copy of dad's chromosome 1 and mom's chromosome 2. Right? At this point, we've now generated 4 haploid gametes. Each gamete is going to have 1 copy of each chromosome. Right? So this could be either the maternal copy, the copy that came from the mom, or it could be the copy that came from the dad. All right, so our end product is going to be 4 cells, and each of those 4 cells is going to be haploid. All right, so this is the overall picture of the process of meiosis. We're going to start with a diploid cell that then undergoes DNA replication, and then 2 rounds of division. The first round is called meiosis 1 and generates 2 cells after the homologous chromosomes separate. And then the second one is going to be meiosis 2, where the sister chromatids are going to separate. At the end, we're going to have generated 4 haploid gametes at the end of the process. Okay, I want to spend the rest of the time talking about each of these different phases in detail. So what exactly is going to happen in the process of meiosis? All right, so the process of meiosis 1 can be split into these phases: prophase, prometaphase, metaphase, anaphase, and telophase/cytokinesis. This is just like what we saw in the process of mitosis. So it's divided into the same phases again. All right, in many ways, the actions are going to be similar between the phases, but meiosis 1 has a few special tricks for this process, or a few special things to take care of. All right, so in prophase, a couple things happen. First, the chromosomes condense, so that means that the DNA packs better around those histones, and the DNA packs together a little bit tighter. This makes it a lot easier to see under the microscope. So when we're looking under the microscope and we can see the DNA in those little stick-like structures, that's because the chromosome has condensed. The second thing that happens that's really very special to meiosis is that these homologous chromosomes pair up and they form a bivalent or a tetrad structure. Right? So what that means is that as they pair up, so as the chromosomes condense inside of the nucleus, you've got chromosome 1 from mom, and that's going to pair up right next to chromosome 1 from dad. So much so that all of the genes that are along these chromosomes, like the eye color genes that I was talking about earlier, all of those genes are going to be paired up immediately and right next to each other. This is really, really important for um, meiosis, because as these chromosomes pair up like this, then they're able to do the next important step, which is called crossing over. All right, so what's going to wind up happening is that as these chromosomes all line up with each other, we've got the eye color gene from mom next to the eye color gene from dad. And as we zoom in and look at these different chromosomes, they're going to switch DNA along that single strand or along that double strand, so that the DNA winds up swapping or recombining from one chromosome to the next. Okay, so here's what that bivalent or tetrad structure looks like. Another name for it is the synaptonemal complex. And in this structure, the chromosomes line up right next to each other. They have proteins that help hold them together like this. And then they're able to do this event called crossing over. So they're going to exchange genetic information between one chromosome and the next. So now we've got mom's chromosome here. This copy, we can trace it from one end to the other. It's now crossed over with dad's, and so it switches from being red to blue at that position. Dad's chromosome does crossovers too. So if we follow this chromosome here, it goes from blue to red, and then it goes back to blue again after that crossover event occurred. All right, so this crossing over is absolutely required for meiosis. Without it, the cell actually cannot continue past this prophase phase of meiosis. The major point or the major purpose of this crossing over event is to increase genetic diversity. It's now scrambling or rearranging the genes on the chromosomes, so that as the chromosome then gets passed to the next generation, the chromosome isn't exactly identical to the one you got from mom. It's got a little bit from your mom and a little bit from your dad, so that your then child, so your next generation or the offspring in the next generation, can be a little bit mixed of both of your parents. Okay, so as a result of crossing over occurring, we're going to wind up with a cell that has chromosome 1 that's mostly mom, but has a little bit from dad. And then the versions that are mostly dad, but then with a little bit from mom. Chromosome 2, the same thing is going to happen on chromosome 2. So we're going to get a mostly dad copy, a mostly mom version. Right. These recombination or crossing over events are actually a little bit more evenly spread out than what I've drawn here, but this is going to make it a lot easier for us to keep track of it. Okay, so this crossing over event is going to occur here in prophase 1 of meiosis, and I marked it in purple because it's one of those very special things for meiosis, and it's really important for generating genetic diversity. So I wanted to point it out, and I wanted that to stand out. Okay, a couple other things are going to happen in prophase as well. Another one is that the meiotic, in this case, the meiotic spindle is going to start to form. Also, the nuclear membrane is going to start to break down, just like it did in prophase of mitosis as well. So let me draw that on here. So here's our nucleus. It is not to scale, but that's okay. All right. Prophase can last a really long time, especially in humans. So in males, prophase lasts for about a week. So as males are making their new sperm cells, it takes about a week to go through this prophase part of meiosis, where all of the crossing over and recombination is going to occur. For women, the prophase 1 of meiosis can last somewhere between 50 and 40 years or so. So when you're born, you've actually got all of your egg cells in you. They just haven't gone through meiosis yet. So every cycle that you're going to experience for the rest of your life is going to send one egg cell through the process of meiosis and generate one gamete for each cycle, each monthly cycle. Those cells, before they go through that monthly, or before they go through meiosis, all of those gametes, all of those gametic stem cells are present in this prophase phase of meiosis. So prophase in women can last for, I don't know, 15 years or however old you were when you first got your period. So maybe, I don't know, 12 or 13 years, or up to as long as like 40 or 45 years when you stop going through meiosis every month. It's kind of fascinating that all of that is just hanging out inside of you, waiting for the next cycle to go through. All right, our next phase is going to be prometaphase. So prometaphase is going to be very similar to what happens in mitosis. So in prometaphase, the nuclear membrane is going to start to break down or finish breaking down. It started a little bit in the prophase part. The pairs of homologous chromosomes are going to attach to the spindle at the kinetochore. So the pairs of homologous chromosomes as tetrads are still stuck together, and they attach to the spindle still stuck together by the spindle grabbing onto the kinetochores of these chromosomes. And this is where the tug of war starts, where the spindle starts pulling back and forth in both directions in order to help separate and divide up these chromosomes. Right. The next phase is going to be metaphase. So now as we go into metaphase, the homologs are going to line up at the metaphase plate. So what this means is that now we've got our cell, we've got the spindles at both ends, and then we have chromosome 1. And so we've got chromosome 1 and chromosome 2. They have been attached by those spindle fibers to the kinetochores, and they're going to line up at the metaphase plate. There are a couple things that I want to point out as being special about this. The first one is that the sister chromatids are still together. Additionally, the homologous chromosomes are still paired together. Right, so here in metaphase, the sister chromatids, the two that are the same color, and the homologous chromosomes, the ones that are different colors, are still paired together. This tetrad formation, or this bivalent, is what has lined up at the middle of the cell. But the other thing I want to point out is that you can get a whole bunch of different types of arrangements for these different chromosomes. For example, for chromosome number 1 here at the top, you could either have mom on the left and dad on the right, or dad on the left and mom on the right. And the chance of either of those happening is exactly 50%, and it's just random chance. On the bottom, for chromosome 2, you could have the same thing. Here we have dad on the left and mom on the right, but we could just as easily have mom on the left and dad on the right. All right, so there are many, many, many different possible alignments or many different arrangements. And the more chromosomes you have, the more different possible alignments or the more different setups you can have. So this process is called independent assortment. And there are many, many different possible alignments, and we can calculate that. So let's say just for this setup here, we could have either mom on the left for chromosome 1 or dad on the left for chromosome number 1. So there are two possible arrangements. All right, on the bottom one, we have the same setup. We have two possible arrangements. So what we're going to have to do is take 2 and put it to the power of the number of chromosomes we have. So here it's 2 to the power of 2, or 2 squared. All right, that's going to equal 4. So for this setup, we've got 4 possible arrangements. All right, so the general equation is 2 to the n. All right, and that's going to equal all of the possible arrangements. For us humans, it would then be 2 to the 23rd because we have 23 different possible chromosomes that we could have. I don't know if you can do this off the top of your head. I can't, so I had to calculate it out. And the answer is 8,388,608. So each of us humans, each individual person could make a possible egg or sperm cell with a different setup or a different arrangement, just based on how the chromosomes are going to line up at metaphase. So we could each make 8,388,608 unique individual cells that all have different genetic information. I think that's so cool and like super fascinating. Like how is it so amazing that we could have so much genetic diversity just based on this feature of independent assortment. I'm going to make independent assortment purple as well because this is a second important special feature that only takes place during meiosis. Mitosis did not have this cool process. So it can move on to anaphase. Now that we've lined the chromosomes up in the middle of the cell, the next thing that we need to do is pull apart or separate these chromosomes. Okay, so here in anaphase, the homologous chromosomes are going to segregate and they're going to move apart to opposite poles of the cell. So here during anaphase, we have the homologous chromosomes separating and segregating and moving apart to opposite ends of the cell. Here they're being pulled by the spindle fibers to the separate poles of the cell. So now we have mom's chromosomes heading to the left and dad's chromosomes heading to the right. All right. Again, important thing to note is that the sister chromatids are still paired together. So the 2 copies of mom's sister chromatids are still paired and dad's sister chromatids are still paired as well. Okay, our last phase is going to be telophase or cytokinesis, and here is where we're going to get the cell separating into 2 separate cells. All right, so I'm going to put mom's or the left-hand cell up at the top. Okay, so here we have our 2 separate cells. The cell on the left is the one pictured here on the top. You've got mostly mom's chromosome 1 and mostly dad's chromosome 2. On the bottom, you've got the blue chromosome 1 and the red chromosome 2. Now, during telophase, all the basic telophase stuff is going to start to happen. So the nuclear membrane is going to reform. Cytokinesis is going to happen at the same time, and we're going to get the actual cell division, and the cell is going to divide completely into 2. So that's our final phase of telophase and cytokinesis. So at this point, we've made it all the way through meiosis 1. Prophase, where the homologous chromosomes pair up. Prometaphase, where they attach to the spindle fibers and start to break down. Metaphase, where they line up in the middle of the cell. Anaphase, where they get pulled apart. And telophase and cytokinesis, where they divide and split into 2. So here is meiosis 1. All right. Meiosis 2 is going to look very similar to meiosis 1. It's going to have the same phases: prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis. However, this time, the sister chromatids are going to separate from each other rather than the homologous chromosomes. All right, so let's look at what that's going to look like. All right, so immediately after telophase, we're going to go back here to prophase. And this time, when we're in prophase, we're going to have 2 cells that we're working with. Again, the same activities are going to be happening here in prophase. So the chromosomes are going to condense and the spindle is going to reform. Prophase 2 is going to look much more like prophase of mitosis because you're not going to get the homologous chromosomes pairing up. In fact, there aren't even homologous chromosomes anymore to do that pairing, and we're not going to get any additional crossing over. So it's going to be very simple and very straightforward, much, much more like what we saw in mitosis. So here's prophase. Our second phase, prometaphase, is going to be very similar to what we saw before. Again, the nuclear membrane is going to break down. And the sister chromatids are going to attach to the spindles. The third phase of meiosis 2 is going to be metaphase. Metaphase is going to look very similar again. Still, we have two cells like this. Our sister chromatids are going to line up at the metaphase plate, and this lining up in the middle of the cell at the metaphase plate is going to set us up for anaphase, where those same sister chromatids are going to get pulled apart. I want to make one important point about these sister chromatids is now, because we've had that crossover event, these sister chromatids are no longer identical to each other. They were identical before the crossover event happens, but now that that crossover event has happened, the sister chromatids are no longer identical. All right, so after anaphase, we're going to get our very last step, or our very final phase of this process, telophase. Telophase is going to happen at the same time as cytokinesis. And at the end of telophase and cytokinesis, we're going to have divided into a total of 4 cells. Each of these 4 cells is going to be haploid. Each one is going to have 1 copy of chromosome 1, and each cell is going to have 1 copy of chromosome 2. So by the time we finish telophase and cytokinesis, we're going to generate 4 haploid gametes, or 4 sex cells, 4 eggs or 4 sperm cells. All right, an important note about these is that all 4 are unique. They're all unique because we've done a whole bunch of things to generate genetic diversity. First, during prophase, we had a crossover event that allowed us to shuffle the DNA between the different chromosomes. Then in metaphase 1 of cell division, or metaphase 1 of meiosis, the chromosome lined up randomly at the middle of the cell. This is called independent assortment and allowed us again to shuffle and rearrange these genes randomly. And then finally, during metaphase 2, the chromosomes lined up again randomly at the metaphase plate, this time the sister chromatids, allowing us to randomly segregate the different sister chromatids at that point. Now, because the sister chromatids are no longer identical, we can have 4 distinct, unique haploid gametes at the end. So now if these are sperm cells, we would actually generate 4 unique separate sperm cells. If these were egg cells, only one of these would be the egg, and the other 3 cells would be helper cells that actually wouldn't be fertilized in order to make the offspring, but they would sort of accompany the egg, at least initially, and would not be fertilized. So only one of these 4 would be that egg cell. So the final thing I want to talk about, and the final thing I want to point out, is about all the genetic diversity, all the mechanisms for genetic diversity that So the last and final thing that I want to point out is all the things that help lead to the genetic diversity that we generate through the process of meiosis. So the first one is this crossing over event. So the first one is this crossing over event. See if I can highlight it. That's not dark enough. Is this crossing over event that takes place during prophase 1. So the first thing to generate genetic diversity is crossing over during prophase 1. The second thing to generate genetic diversity is independent assortment that takes place during metaphase 1. And then again during metaphase 2, when the sister chromatids line up at the metaphase plate again, we get another round of independent assortment at that point. So the final product that we generate are these 4 separate unique haploid gametes. All right, all of this is a result of the genetic diversity that is put into the system through the process of meiosis. All right. I hope this was helpful. If you have questions, please let me know. Bye.